everyone. So today's experiment is to study the population density of different plant species. Okay. So we have a population density chart which is carried out by the quadrant method. Okay. So quadrant means a species which present in the four lines, that is quadrant. Okay. So here the formula of quadrant is D is equal to N by S. So D is considered as population density, N is considered as number of individual, and S is considered as unit of space. Okay. So now mass by volume is our formula, which is N by S. Okay. So now total space of any area of farm or soil, that is mass. Total occupied space, that is volume, is V. Okay. Now population density is carried out by some chart. So what is population density now? Number of individual species present per unit area of a given time called as population density. So how we can calculate it? So we have some plant species, which is 5 plant species, having different size, shape or color. Okay. So now this is our chart of population density. So it is shown by different colors and different shape of the leaf, flower and etc. Okay. So species A, B, C, D and E is shown here. Okay. So we can take some quadrant row. Okay. So here row 1, 2, 3, 4 and 5 is there. Okay. So in case as an example we take row 5. Okay. So in row 1, the 1, 2, 3, 4 and 5 quadrants are there. Okay. So number of quadrant is 5. Okay. So every species is present in every quadrant. So your number of every quadrant occurred species is also 5. Okay. So in example we can take 5 quadrant. So first quadrant you have to take and the number of individual per quadrant you have to calculate. Okay. So this is your quadrant number 1. Okay. This is your quadrant number 2. So you have to calculate per species and write in your observation table. So observation table for the particular 5 quadrant is, first row is serial number, second row is plant species A, B, C, D and E. In front of them the size and shape of that particular plant species is there. Now number of individual per quadrant is 1, 2, 3, 4 and 5, which is our row 1. Okay. Now in chart row 1 is 1, 2, 3, 4, 5 quadrants. Okay. Total number of individual in all quadrant, that is N. Next thing, total number of quadrant species occurred, that is A. Total number of quadrants studied, that is B, and population density formula, that is N by D. Okay. As per mass by volume. Now we have to calculate the first quadrant, that is first species. Okay. So you have to count the first structure in the particular chart. Okay. So first structure is 1 and 2. So you have to write 2 here. Okay. In second quadrant there is only one structure. So now you have to write 1. Third quadrant, the structure of species A is 2. You have to write 2 here. In fourth and fifth column there is no species A. So this is your 0. Now total number of individual in all quadrant, it's summation of all number of individual. Okay. So you have to summation it, that is 5. Total number of quadrant species occur is in only 3 because the fourth and fifth one is 0. So 3 is your count. Okay. Total number of quadrant study is 5. In all rows there is a 5. Okay. Because we studied only 5 quadrant here. Okay. Now population density is calculated by N by V. So your N is 5, V is 5. So 5 by 5 is equal to 1. So you have to calculate per quadrant here in observation table.